Good afternoon, everyone. Today we will be discussing about Mendelian genetics. So, Gregor Johann Mendel, he was called as a father of genetics. So, because he has given some laws to explain the genetic inheritance in human beings and plants. Okay, so how basic inheritance of genes happens in the organisms, even in the animals. But of course, there are some non-Mendelian inheritance also, which is available. That also we will discuss. So Gregor Johann Mendel, so he was responsible for the laws governing inheritance of traits. So actually, out of his own interest, he has done some experiments and he has done some calculations to derive some laws. So which explain how the inheritance of genes occurs in the organisms. But this is true universally. That is, in most of the cases, you can actually see this kind of inheritance. So he was an Austrian monk. So he studied the inheritance of traits in pea plants, and he developed the law of inheritance. And when he was doing this experiment, the main thing is for his success is the experimentation. So that is very much important. So the correct sample, the correct model for the experiment he chose, and everything he documented. So he has not left anything. So he has documented each and every result, and then properly he has maintained. So because of that, actually he was successful in deriving the laws. But during that stage or during that period, his laws were not accepted. So because of various reasons, like some will have the religious or some already well-developed ideologies. So because during those times, so they used to think that, okay, God has created, okay, that is another aspect. But when it comes to science, so when there are some laws, so which can be proved, so that has to be accepted. But that was not the ideology during that time. So there is some spontaneous theory of spontaneous generation. That means everything came spontaneously. Okay, there was no pre-existing cells. Like that, there are different theories. So because of which they could not accept his theory of inheritance. So until 20th century, his work was not accepted. Later, some scientists from different countries, they independently, actually they were searching for something in the library, and they could find his paper published. So they could find that paper, and they have studied and they calculated again. So they got the same result. So that is a very good experiment. That means reproducibility is very important in doing any experiment. That means if I do one experiment, I am getting some result. If another person is doing the same experiment, he or she also should get the same experiment, same result. Then that experimentation is correct and the results are accurate. So therefore, they could also do the same kind of calculations and they got the same result. So then they were like interested, like, oh, this is wonderful. But by the time, by the 20th century, a lot of advancement has already happened in cell biology, molecular biology, then DNA, all these were already started. So therefore, then actually his contributions came into light, and then his work was appreciated after 100 years of his death, almost. So between 1856 and 1863, Mendel cultivated and tested some 28,000 pea plants. And he observed that the trait means the phenotypes, or the the characters. Traits means characters. That means a height, tall, short, curly hair, long hair. All these are different traits. So children usually carry the traits from their parents. So that is what he observed. So this is the site where he performed his experiment. Now it is in the Czech Republic. So what he actually, he don't know all these genes, all these, he doesn't know the terms genes or DNA. All these were later, after his death, they were identified and named. So during that time, Mendel, he stated these physical traits are inherited as particles. He told that physical traits, that means though we have genes, until they are expressed and we are getting some physical appearance, whether okay, tall, short, all these, they are coming as a particles. So he did not know what are these particles, but later actually they were chromosomes and DNA. So these are the basic genetic terminologies you should understand. Trait means any character that can be passed from parent to offspring. Then heredity means passing of traits from parent to offspring, that is heredity. Genetics means the study of this heredity is called as genetics. Then we have different genetic crosses, monohybrid cross and dihybrid cross. So monohybrid cross means cross involving a single trait. That means flower color, height, pod shape, flower position. Okay, or any of this, only one character it is. Dihybrid cross means cross involving two traits, flower color and plant height. How these two means we will get different combinations. Then all these calculations were performed using a square which is called as Punnett square. Even now we use this Punnett square to solve these genetic problems. So how to draw the Punnett square is nothing but, so here first we have to draw the square box, and the boxes depends on the, like whether it is monohybrid cross or dihybrid cross. So here we have to make a grid, place the alleles of the gametes. So here we all know that there are 2 chromosomes in each organism. So one comes from the father and one comes from the mother. So that is called as allele. For if you take each gene, suppose height of a person. So tall or short. So one allele will come from the mother and one comes from the father. When these two combines, that is called as genotype. Okay. And these are called as alleles, each type. So then there will be different combinations. So how the combinations comes, that we will see. Suppose here you see both parents have LOP allele, that is capital Y, small y. So Y codes for yellow color, capital Y. Small y codes for green color. But here capital Y is the dominant one. So though you have small y, only the P color is yellow. So here you have both the parents have capital Y, small y. Therefore, if you draw a box, first you have to, on the x-axis, that is on the top, you should write parent 1. On the left, you have to write parent 2. So capital Y, small y, and here also capital Y, small y. Then we have to fill the grid with the combination. That is, suppose if the capital Y from one parent and other capital Y from the other parent, then the offspring will get capital Y, capital Y. Then another offspring may get capital Y, small y. So again, the bottom also, from second parent small y and first parent capital Y. Okay, capital Y, small y and small y, small y. So that means there are a chance of getting any of these 4 combinations. Okay. So as I told you, capital Y, that is yellow, is dominant than the small y, which is green. Now, when there are 2 copies of capital Y, definitely the P will be yellow in color. So here you have to observe one thing is, you will get genotype ratio and phenotype ratio. So genotype means what is the combination of alleles. Okay, that is genotype. Phenotype means expression, that means character. Now you see here in the second box, you have capital Y, capital Y, capital Y, small y, small y, small y. There are three combinations. So this is called genotype. So how I have to write the genotypic ratio? So each combination, how many number of organisms are there or offsprings are there, I have to write. So capital Y, how many of there? 1 is to capital Y, small y. How many? 2. So 2 is to small y, that is 1. So 1 is to 2 is to 1 will be the genotypic ratio. When comes to phenotypic ratio, phenotype is nothing but the expression. As I told you that Y is dominant, capital Y is dominant, though the capital Y is present in 2 copies or even a single copy also, the P will be yellow in color. So capital Y gives yellow, capital Y, small y also gives yellow. So capital Y, small y, you have 2 copies, right? So out of 4 offsprings, 3 offsprings will give you yellow color and only 1 offspring, that is, if both the alleles are small y, then the P will be green in color. So how I have to write the phenotypic ratio? Yellow is to green. So yellow how many? 3. 3 is to 1 green. Okay. 3 is to 1. So this is the phenotypic ratio. This is a simple monohybrid cross. Even if you write for dihybrid, trihybrid, whatever it is, in the same way you have to write the ratios, genotypic ratio and phenotypic ratio. I hope it is clear. You have to practice. So at the end of this slide, you have some practice things. So which I will explain 1 or 2. You have to practice. Then it will be very easy and it is very interesting also. So this is called Punnett scale. Then genes. So in the genes, the terms are like alleles. Alleles are the 2 forms of a gene. So here one will be dominant and other will be recessive. So dominant is nothing but stronger of the 2 genes expressed. So it is usually represented with the capital letter. That means even if 1 copy is there also, you will get the same character. Recessive means the genes that show up less often and only when 2 copies are present, only then it will get expressed. So that is called as, that is expressed in the lower case. That is represented in the lowercase letter. Then genotype, which is the genetic combination. So here you can write RR, capital R, capital R. That is called as homozygous dominant, because R is dominant and both are R, capital R. So you call homozygous dominant genotype. If both are small r, then homozygous recessive genotype. And if it is capital R and small r, then you call them as heterozygous genotype. Okay. Then phenotype. So phenotype is the physical feature resulting from the genotype. That means what is the representation? We don't know what is the, like if you see, suppose I am tall. It may be capital T, or capital T, small t. You just this is for our representation. You don't see capital T, small t on our chromosomes, right? So we don't know actually what allele or this. It is only the appearance that is present. So that appearance is called as phenotype. So if capital R is present, or even capital R, small r is present, the flower expresses red color only. And if small r is present, then the flower expresses white color. So this is called as phenotype. So here, this is how it looks like in the genotype and phenotype in flowers. So genotype of alleles, that is capital R gives red color, small r gives yellow flower. So all genes occur in pairs. So 2 alleles they both will affect the character. So here, if capital R genotype is the pair, that is homozygous dominant, phenotype will be red color. Heterozygous, that is Capital R, smaller. You should not call heterozygous dominant or heterozygous recessive. So because heterozygous itself say that one dominant and one recessive allele are there. Okay. You should tell dominant or recessive only when you are telling as homozygous. Whether it is both capital R or both small r, like that. Okay. So in the heterozygous condition, usually it will be red because R is dominant, capital R. Then if both are, that is homozygous recessive, it will give yellow color. So here homozygous genotype, heterozygous genotype. So homozygous genotype is nothing but gene combination involving two dominant or two recessive genes. So this is called as pure combination. Okay. Heterozygous is nothing but the gene combination of one dominant and one recessive allele, and it is called hybrid because it is derived from both the parents. So not only genes, here the characters are also influenced by the environment. Okay. So here, how he performed this P experiment that we will see. So the scientific name of the P is Pisum sativum. So the best, what is the reason for the success of his experiments was that he has selected the perfect model for his experiment. Because this Pisum sativum, it can be grown in small area. See, if an individual has to do research, so it's very difficult. Now, if you want, suppose animal models or anything, you need a sophisticated room where the temperature is controlled, light is controlled. So you have to have some feeding, time to time we have to feed. So it will be costly and lot of investment is required. You have to make sure that there will be no infections, all these things. So here, when he selected Pisum sativum, there is no such complications. They can be grown in very small area, even in our pot. That is the best thing. So he has actually grown them in his backyard. Second is, it produces lots of offsprings. So when you have lots of offsprings, we can actually find the variations. Okay. For example, I am doing experiment on some big tree. Okay. Or some animal. So what happens? Suppose higher animals, they will usually produce one or two offsprings. Suppose white color guinea pig. It will give both white color guinea pigs only, offsprings. Okay, then what is the use of doing such experiment after some 1 year or 2 years? It is giving only one or two offsprings. I'm just giving you example. Don't take it literally. So what I'm telling is, if you are taking some animal or some other plant or tree, which usually give less number of offsprings, then what happens? The variation, the chance of variation or expression of genes will become limited. Okay. So therefore, you cannot get actual results here. Okay. So here, but Pisum sativum, it gives lots of offsprings. Therefore, you can get lot of variation here. And they produce pure plants when allowed to self-fertilization, self-pollination. So that means tall plants will give only tall, short will give only short. So that is one chance. This is by self-pollination. And if cross-pollination is done, then you will also get like hybrid varieties also. So you can actually do your kind of experimentation also. So that is very important. So how the reproduction in flowering plants takes place? Here, the pollen grains, actually they contains the sperm. So at the end tip of the stamen, usually pollen grains are present, and ovary contains eggs which are found inside the calyx. So here the pollens carry the sperm to the eggs for the fertilization. And self-fertilization will happen within the same flower. Cross-fertilization occurs between the flowers. So actually what he did, because the flowers are very small, actually he could pick up the flowers, he could hand pollinate with a paintbrush. He actually took a paintbrush, he took pollen grains, he actually fertilized with the other plants. He could snip the stamens to prevent the self-pollination also in such plants. And he covered each flower with a cloth bag. Then he traced several traits for several generations. And one more important thing is, it will give lot of offsprings and the generation time is also less. For example, I am telling, if you are doing experimentation on elephant, like after 2 years, actually it will give the offspring. Suppose you are doing some experimentation and after 2 years, you are not getting the desired result. So what happens? It's a waste of time, right? So you should never select such kind of models. Okay, you should always select the models, whether it is plant model or animal model or microbial model, which has less generation time so that you can easily see the result within no time. And if you are doing anything wrong, you can rectify. But if you are doing in such big animals or trees or something which have very long regeneration time, what happens after 2-3 years of spending of your career and lot of money, if you are not getting the result, then it's of no use, right? So that is very important. So Mendel produced pure stains by allowing the plants to self-pollinate for several generations. Then he has selected 8 pea plant traits. And one more reason for his success is, so here for each trait, there were only 2 variations. That means if the seed shape, it is either round or wrinkled. But he did not select such characters which have a range. That means, suppose I am taking the color. Either it is green or yellow or green. That's it. Only 2 colors. Not yellow, green, red, white, different combinations. If such kind of character we have taken, you will get confused what it is getting. So that is the main reason for the success. So he selected 8 pea plant traits, that is seed shape, seed color, pod shape, pod color, seed coat color, flower position, plant height, and flower color. So what are the combinations? That seed shape is either round or wrinkled, seed color is yellow or green, pod shape is smooth or wrinkled, pod color green or yellow, seed coat color gray or white, flower position axial or terminal, plant height tall or short, and flower color which is purple or white. So all capital letters are dominant, represented in the capital letters. That is, in the seed shape, round is dominant over wrinkled. Okay, like that. So this is how it looks like. So this table is important. Seed shape, smooth, wrinkled. Smooth is dominant over wrinkled. Seed color, yellow is dominant over green. Pod shape, inflated is dominant over wrinkled. Pod color, green is dominant over yellow. Flower color, purple is dominant over white. Flower position on the stem, axial is dominant over terminal. Then stem length, tall is dominant over dwarf. So like that he could get. So once he has done, so here in the F2 generation, so F1, F2 generation is nothing but after first cross, what are the offsprings he is getting, that is called as F1 generation. And on the second cross, that is after crossing the 2 hybrids, whatever the offsprings getting, they are called F2 generation. So this is the ratio he got. So for each one, so smooth to wrinkled, so 2.96 is to 1, like that he got. So did the observed ratio match the theoretical ratio? So the theoretical ratio or expected ratio of the plants producing round or wrinkle is 3:1. So what will, just now I have explained you the Punnett square, 3:1. So 3 dominant and 1 recessive we will get. That is a theoretical. But what in the practically he got is 2.96:1. So this is different from the theoretical. So there are in a digits there is difference. So this discrepancy is due to the statistical error. So when you take the large sample size, whenever you are doing the experimentation, if you take large sample size, you usually get the approximate result which is near to the theoretical value. So now what is F1 and F2 generation? So here parental or P1 generation is nothing but the parental generation in a breeding experiment. So that is a starting generation. F1 generation is the first generation or called filial generation. So it's offspring in a breeding experiment. So F2 generation is the second generation offspring. This is called second filial generation. So in the first step, parental generation, so pure tall he took and pure short. That is capital T to small t, small t. When he crossed all the offsprings, because see, if suppose if one from the tall plant, only we will get tall allele, T allele, capital T allele. And from the short plant, we get only small t. So all the offsprings will get one copy of capital T and one copy of small t. So all the offsprings of F1 generation, they are hybrid. And they are all again tall because capital T is dominant over the small t. Now in the F2 generation, this hybrid is crossed with another hybrid. That is capital T small t. A plant having capital T small t is crossed with another plant which is having capital T small t. Then what happens? We will get different combinations. I told you like Punnett square. After drawing the Punnett square, one offspring will get both copies of capital T. One offspring will get both copies of small t. And two offsprings will get one capital T, one small t, and one capital T, and one small t. So here the genotypic ratio is 1:2:1. Whereas phenotypic ratio is 3 tall and 1 short, that is 3:1. They are called monohybrid cross. So this is another example. So here P1 monohybrid cross, where this we are taking in seed shape. Alleles are capital R is round, small r is wrinkled. So the cross is between round seeds and wrinkled seeds. So from capital R and small r, small r. So from capital R, the two alleles will segregate. Capital R, small r. Capital R, capital R. From RR, small rr, both the alleles will segregate. And we will get the combination of capital R, small r for all the 4 offsprings we are getting. 4 means here not the number. Like you consider as a whole. So 1/4, 2/4 like that. Okay. So here the genotype is capital R, small r, phenotype is round. So everything is all have same kind of phenotype. Now this homozygous dominant is crossed over homozygous recessive, and offspring will have the heterozygous hybrids, that is F1 generation, and genotypic and phenotypic ratio is same for all. Then in the F1 generation, capital R small r is crossed with another hybrid, which is capital R small r. So when you draw the Punnett square, we will get 1 capital R, 2 capital R small r, 1 small r, and phenotypic ratio is 3 red and 1 white. Oh, sorry, here round, Here 3 round and 1 wrinkle. Okay. So this is how the ratio happens. So here that is 25% are homozygous dominant, 25% are homozygous recessive, and 50% are heterozygous. Okay. So this is how the pea looks like texture, all this. This is the real-time image. Then we have another terms like test cross, back cross, all this. So test cross is nothing but, so this F2 generation, whatever he got, that is hybrid, capital or smaller. He crossed again with the pure combination. That is either homozygous dominant or homozygous recessive. So like this, capital R with capital R smaller, or smaller with capital R smaller. That is one hybrid and one homozygous, whether dominant or recessive. So in such case, what are the different combinations? So from capital R, definitely we are getting 2 copies of capital R. From capital R smaller, we are getting 1 copy of capital R, 1 copy of smaller. So here you can see, yeah. So here, so I will show you. So here you will get capital R, capital R smaller. That is the combination. Then again in the bottom also you are getting capital R smaller. So that means genotypic ratio, you have capital R, how many? 2. Capital R smaller, how many? 2. That means you can just write as 1:1 ratio. Okay. So you should not call 2:2. Okay. 1:1 ratio. Both are equal. 50% capital R, 50% capital R smaller. Whereas phenotypic,
one is homozygous, one is heterozygous.
Sorry?
In genotypic ratio, one is homozygous and one is heterozygous, yes?
Right. Yeah. So 50% you are getting homozygous dominant. 50% heterozygous. So 1:1 ratio. That is genotypic ratio. So in the phenotypic ratio, how we are getting both, whether you have capital R or capital R smaller, you are getting round only. So all 4 will get round seeds only. Okay. All alike. So 100% will get the round seeds. So this is called test cross. So same thing with the recessive, homozygous, recessive, and hybrid. So here what you are getting, 2 homozygous offsprings and 2 heterozygous offsprings. Homozygous recessive offsprings are 2, heterozygous offsprings are 2. So here the genotypic ratio is 1:1 again. Capital R smaller is to RR, smaller. When phenotypic ratio, if you see, capital R smaller will give you round seeds, smaller will give wrinkled seeds. So round to wrinkled, you are getting how many? 1:1 ratio. That is 50% of the offsprings will get round seeds, 50% of the offsprings will get wrinkled seeds. Okay, this is phenotypic ratio. So why it is called test cross means, so to check whether the offspring is having the same genotype as that of parent or not. Okay. So that is 50% of the heterozygous and 50% homozygous. So the ratio should get 1:1. So when you perform this experiment, this result or this calculation, you should get same or similar ratios. Then it is correct. Okay. Now these are some practice crosses are also there. I will show you. So now what are the results of these monohybrid crosses? So now what are the outcomes of his experiment are not only this ratio, that is the genes or whatever he said particles. So the inheritable factors or genes are responsible for the heritable characters. And phenotype is based on the genotype. So only based on the genotype, the characters are expressed. And each trait has, that is, suppose height. It has 2 genes, tall, short, like that. One comes from mother, other comes from the father. So true breeding means homozygous. Both the alleles will be same. Then the heterozygous is the hybrid. So law of dominance and law of independent assortment. These are the Mendel's laws. So first law of dominance means in a cross of parents, that the pure for contrasting pairs, that is, suppose if one father is tall, suppose he is having capital T, and mother is short, small t. They are pure. That means father is not having capital T, small t. Okay, he is having, this is the assumption I am telling you. So capital T is having, and mother is short, she is having capital small t. Then they are pure in their contrasting characters. Okay. So when comes the next generation, usually they will be heterozygous. That is, from father capital T is coming, mother small t is coming. So the offspring will have now capital T small t, which is heterozygous or hybrid variety of genotype. But here, still he or she is tall. This is because of dominant allele or dominant trait. That means capital T is dominant. That's why even though small t is present, still the offspring is tall. Okay. So like that, you can take any of the examples. This is called law of dominance.
So, ma'am, in law of dominance, only one gene will appear. One is the dominant in offspring.
Yeah, one allele will be dominant over the other.
Okay.
Okay, we have two alleles in any gene. So one will be dominant. That is masking the expression of other. Okay, so only one will be expressed. If you see the, suppose height, only tall will, a single person cannot be tall and short at the same time, right? So
Yes,
only
ma'am.
one will be expressed, or long hair, short hair, or curly hair or straight hair. Like that, you can take fair or dark complexion, anything. You can take any example. Just I'm telling you. So any thing, only one will be expressed and that will be dominant. If both the copies are having same allele, then obviously that will be expressed. But when there is a heterozygous combination, then which allele is expressed, that is called as dominant. And other will be called as recessive. Is it clear?
Yes, ma'am.
This is law of dominance. But even there are some non-Mendelian inheritance also where both will express. That also we will discuss in the next class, or if time permits today only. Then law of segregation is the two, during the formation of gametes, that is eggs or sperm, the alleles will be responsible for trait will get separated from each other. So that means, see, if a man is having X and Y, right? If a female, she's having both X chromosomes. But when it comes to the next generation, only X or only Y from the father should come to the baby, but not both X and Y from father only and nothing is coming from mother. It is not that case. So either X or Y should come. And from mother also, only one copy of X will come. Why it is happening? For that, first these two has to separate. Okay. So they should not get linked. Okay. So now if you see in each sperm, only one allele will be present or one chromosome will be present. That is either X or Y. So which sperm is fusing with egg, depending on that, the gender of the baby will depend. Okay. And in the egg, either of the X copies will be present. So this is called as law of segregation. That is, they have to separate from each other. And they will get combined after fertilization, and that will actually give the combination for the offspring. And based on that, based on all the combinations of different genes, all these phenotypes will be expressed in the baby. But here, so this is how female parent, these two gametes will separate, eggs. And male parent also they will separate, and different combinations will form, and based on that, the expression of characters will occur in the baby. Height, weight, color, everything. Okay. Not only height, weight, color, these are what we see outside. Every enzyme, protein, everything will be expressed by the genes, right?
Yes, ma'am.
So then law of independent assortment. That means these two are independent of each other. Okay. That means there is nothing like they are dependent on some other. If a capital T is there, then definitely you should get small t along with that. No. Usually in dihybrid process, if you see if this is capital T small t, we are discussing only the monohybrid process. That means only one character. But in real time, it is not that each gene will come independently, right? Because once the chromosome is replicated, chromosomes, how many chromosomes are there? Once they are replicated, all the genes will be passed to the offspring. So if the inheritance of each gene again depends on the all other genes also, right? So in the dihybrid cross, usually we will get to know this law of independent assortment. That means not the inheritance of one gene is not influenced by the inheritance of other gene. So I will tell you one example. So suppose father is he is tall and he is having straight hair. Mother is short, having curly hair. But that doesn't mean that if the offspring is tall, that he or she should get the straight hair only. As father is having tall, straight hair genes and mother is having short, curly hair, it is not that if the daughter is short, she should get curly hair only. No, she may get straight hair. Or the daughter may be tall and she may get curly hair from mother. That means each gene is independent of the other. Inheritance of each gene or allele is independent of the other. It is never dependent. That is called as law of independent assortment. So each pair of alleles segregate independently during the gamete formation. And how many number of heterozygotes we will get, that is given by the formula 2 to the power of n. So how many number of combinations? Okay. So n is heterozygotes, number of heterozygotes present. Okay, that you can, while practicing, you can see here. Yeah. So you can see simple one I am just explaining. You can practice with others. Capital R, small r, capital Y, small y. So you have to separate capital R, small r, capital Y, small y. Then from the other parent also we will get capital R, small r, capital Y, small y. Then we will get different combinations. That is capital R, capital R, capital Y, capital Y, capital R, small r, capital Y, small y. Like that we will get different combinations.
Can you please show like
Yeah, I
how
will.
to do that?
If it is there here, I will just show you. Yeah, so this is dihybrid cross, you can see. So capital R, small r, capital Y, small y. This is, that is, one seed is having round shape, yellow color. Both round shape, yellow color. But they are hybrid. Now when their gametes are formed, they will have 4 different combinations. That is, they may get capital R and capital Y, capital R, small y, small r, capital Y, small r, small y. So I will show you if it, yeah, here. Wait, I will shop. Just a second. So can you see the screen?
Yes, ma'am.
Okay, I'm just trying to. So Wait. Okay, I will share here only. There's some confusion here. Yeah. Okay, I will explain here only, but just understand slowly. So now you understood now here, till here, that so how we have drawn Punnett square. So that means, so here out of capital R small r, first you have to divide that capital R separate and small r separate. Then capital Y separate, small y separate, and you have to combine all the 4 combinations. So you may get capital R, capital Y, capital R, small r, capital Y, small r, small y. Any of 4 are possible. So same with this case. So you have to write in the Punnett square on the top, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Same thing in the left side. Now you have to combine this. So in the first box, what you will get? You can just try. Capital R, capital R, capital Y, capital Y. Right?
Yes, ma'am.
So in the adjacent box, capital R, capital Y, small y. So in the first row, I am telling you. Then in the adjacent box, capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Like that, you have to fill in all the box. So you will get like this. Okay. So now you have to write phenotype and genotype ratio. So first we will see phenotype. It will be easy for you to understand. So capital R is round, small r is wrinkled, capital Y is yellow, small y is green. Just remember that. So in the first box, both are capital R, that is round, capital Y, round, yellow. So round yellow, how many are there? 1, 2, 3, 4, 5, 6. So that is in first row total, there capital R, that is round and yellow. Then in the second row, only 2 are round yellow. Third row, 2. Fourth row, 1. So 4, 5, 6, 7, 8, 9. So 9 will be round yellow. Then round green, that is, it should be capital R and small y should be there. Both the copies of small y. So that is in second row. Second row, 1, 2, 3. So in the last row, 1. Okay. So 3 are round green. Then wrinkled yellow. That means small r, and one should be by capital Y. So that is 1, 2, 3 in the third row and fourth row. So there are 3. Then wrinkled and green, that is only the last one, which is small y, small y. So that is 1. So 9:3:3:1 is the phenotypic ratio. And the genotypic ratio, you have to write for each thing. That is capital R, capital Y. How many are there? Only 1. Capital R, capital Y, small y. How many are there? 1, 2, only 2. Like that, you have to write for each combination. Right? That is a genotypic ratio. This is phenotypic ratio. So this is the test cross. Okay. So you have to practice here. So how, like here something is missing, no? So what should be there here? Like that you have to practice. So this is the test cross result. So this is the summary of Mandela's. So I will just tell the summary and I'll stop here. I will share the PPT in the group today itself. You can practice, and if you have any doubts, you can like get back to me tomorrow. So there are 3 laws: law of dominance, law of segregation, and law of independent assortment. So in the law of dominance, completely, so the dominant allele will express 100%. If the dominant allele is there, that will be expressed. And segregation means these 2 alleles are independently segregated. Okay. that is one comes to sperm and other comes to egg, and they will segregate. Independent assortment means the movement of one allele is never influenced by the other movement of other allele of the other gene. Okay, so these are the three Mendel's laws. So yeah, incomplete dominance and codominance are also present. So in the incomplete dominance, the F1 hybrids have an appearance somewhat in between the phenotypes. That is, domination is not complete. So here you can see example, you should get red. If RR is dominant, they all should get red. And if only smaller is present, then they should get white color. But in the snapdragons, you may get red, pink, white, like that. The pink means it is in between. The heterozygous may give different color, like that. Okay, so here you can see. If there is a capital R, small r, then it is giving pink. That means if both white color, if capital R, small r, then it is pink color. So that is called as incomplete dominance. Like this. The ratios are given here. Codominance means both are dominant. Both will get expressed. Best example is blood type. So the two alleles are expressed. So here in the type A, both the alleles will have A. In the B, you have both B. In the AB blood group, one is A and one is B. So either you should get A or B if dominance, law of dominance is applicable. But here you are getting AB blood group. That means both A and B, both are dominant. And the O, you're not having A or B. Hence, it is called type B, type O. Is it clear? This is codominance. So this is how the cross is. Suppose male type is O. He's not having anything like II. And female is AB. IA, IB. So IAI, IBI, IAI, IBI. So IAI means A. IBI means B. So two offsprings are having A blood group and two offsprings are having B blood group. Okay. That means even father is O, mother is AB, but children are getting either A or B. Okay. Like this. This is the problem. So you have some questions here. You can just practice. Then sex-linked traits are nothing but they are attached to they are present on the X and Y chromosomes. So they are inherited depending on their chromosome. That is mostly sex-linked disorders. That is, if they are present on Y chromosome, only the male child will get affected. Because female, she is not get. Suppose father is having some disorder causing gene on Y chromosome. So that will come to only son, but not the daughter, because daughter is not having the Y chromosome. So that is another. Or if mother or father, they are having X-linked disorders, then female child is having great chance of getting the disease, and the intensity will be more in, suppose if it is dominant. So you understand that if X chromosome dominant disease is there, suppose if even single copy of X is present also, then the disease comes, then the son will be more affected, because even the XY is there, even single copy of X chromosome is present also, he will get the disease. If it is a recessive disorder, then he is having less chance, then girl child is having more chance of getting the disease. So such kind of things can expect in the sex-linked traits. So like this, you can see eye color in fruit flies. So this is the crossing. So here hemophilia is the best example. So women are the carriers. They are sex-linked traits. So men will usually express. So here best example is Queen Victoria. She is carrier. She is having the gene, but she is not having the disease. But her son, sorry, her daughter Elise, then Alexandra, all they were carriers. But after 3 generations, Alex, he was the one, actually he got the Elise because it is recessive. So he got that particular disease and he died of the hemophilia. Okay, so these are various combinations you can actually practice. So do you have any doubts here?
Hemophilia means color blindness.
Bleeding won't stop.
Okay, okay.
Okay, so if there is any cut or something, so the bleeding can't stop. So they will die of bleeding. Okay, so you just practice. No need to go into depth. Only monohybrid, dihybrid cross, test cross. You can just check. Okay, you have different examples, right? You can just practice all, and I will share the PPT now. If you get any doubts, you can come back to me in the next class tomorrow.
Okay, ma'am. Ma'am, you can share the important questions also, ma'am, because zoology syllabus is very, very lengthy, ma'am.
Okay, okay. Okay, by evening or by tomorrow, I'll share. Okay?
Okay, ma'am.
Okay. Thank you so much.
Thank you, ma'am.